This is a video tutorial for the basic navigation of the RiboVision portal. Across the top of the screen, you will find our one-dimension of data panel. We call this feature Navline, as in navigation line, because it displays data within a reference frame that is the coding nucleic acid sequence from 5' on the left of the screen to 3' on the right. This is our main panel, where we have 2D secondary structure maps. And over here is the JMOL panel, where you can see the molecule in 3D. The display of data on 1D, 2D, and 3D panels can be synchronized or visualized independently. On the right side of the screen is a toolbar with the major functional tools for the website that we will discuss later, and on the left side is our main menu. You can find links to the written manual and further video tutorials by clicking here or here.
So the first thing you need to do is load your molecule of interest for your session from the species selection menu. We have Escherichia coli, Thermus thermophilus, Haloarcula maris mortui, and Saccharomyces cerevisiae. For this example, we will use E. coli. Further, in this menu, you may choose either the large subunit or small subunit. We will choose the large subunit. In the future, we may have multiple structure options here. Those will appear at the next level here. For now, we select Load Data to continue.
Upon selecting the map, the 2D panel will immediately load. We have different default views for each panel. You will see the secondary structure with black letters. Up here on the nav line is what we call the rainbow. You can see nucleotide sequence indices from 0 to about 3,000 for each nucleotide. The 3D view of the ribosome loads in the JMOL panel on the right. Please be aware that the JMOL panel may take a minute or two to load. You can see that the default color scheme illustrates the 6 different domains of the ribosomal RNA. You can click and drag to rotate it around.
Now we will explain how to move objects around using a standard 3-button mouse. You can drag the 2D map around with the mouse in the X and Y directions, as well as zoom in with the scroll wheel on your mouse. Alternatively, you can use our compass buttons here in the upper left corner, our zoom bar, and this big letter R, which resets the map to the default view in case you get lost. If you move your mouse pointer to the JMOL panel at the right, navigation control will be transferred to the JMOL app. Note that your mouse navigational controls will operate differently once you move from the 2D panel to the 3D JMOL panel. To rotate the molecule in the JMOL panel, click and hold the left mouse button. To translate the molecule in the plane of the screen, point the mouse to anywhere within the JMOL panel, hold the Shift key, then double-click and hold the left mouse button. Finally, the mouse wheel controls zoom. Scrolling will zoom the molecule in and out. For those of you who do not use a 3-button mouse, please see our advanced navigation tutorial.
From here, we can load data. To do this, click on one of the data sections in the main menu. We have the structure data, alignment data, protein data, and interaction data sections. The data sets we will use as examples in this tutorial are rainbow, where the residues are colored according to their index in the rainbow scheme from blue to red, and domains, where the nucleotides are color-coded according to the secondary structure domains. You can see here a brief description of each type of data. You can also find a link to the relevant section of the written manual with a much more detailed description of the data and relevant scientific citations. From here, the data shown in the 1D, 2D, and 3D panels are synchronized unless you use advanced techniques illustrated in other videos.
In the protein data section, you can choose to visualize the entire set of proteins for a ribosomal subunit, or you can select a desired protein or subset of proteins from the list in the Protein Data menu by checking the boxes. Upon selection of a set or subset of proteins, you can see that on the 2D panel, the colors of the nucleotide letters have changed. Colored nucleotides are nucleotides that are in contact with a protein in the 3D structure. Each nucleotide color in the 2D structure corresponds to a protein of the same color now seen in the JMOL panel.
You can also graph 3D interaction data in the 2D panel, which are shown as lines between nucleotides that are close in 3D space according to various criteria. We will demonstrate this feature by selecting Base Pair Interactions from the Interaction Type menu. Some interaction types can be further characterized into subtypes, so we clear out the Interaction Subtype menu and choose, for example, cis-Watson-Crick base pairs, abbreviated as CWW. You will see that lines appear between some nucleotides. Each line connects two nucleotides, forming a Watson-Crick base pair.
RiboVision also provides options to export or import data. These options are contained in the main menu. The Import menu allows the users to upload their data as a function of ribosomal RNA nucleotide in a table format. The Export menu enables the users to save their artwork. Detailed descriptions of the import-export options are presented in the Advanced Tutorial section. This concludes our description of basic navigation.